Hello everyone, so this is Bassi and I welcome back to this particular channel. So we are going to have our second class of this particular dropper to topper batch. In the previous session we discussed about the mitosis and I hope that it's clear to everyone, and now today in this session we will be going to convert these questions, the questions which is going to come into this from this particular chapter. We are going to convert those questions into a very simple questions, right? So today, before starting the meiosis, I would like to revise all the things along with me. So yesterday, if you have not watched the session, go to back to the that after the class, go back to the videos, watch those videos and let me know if you have any kinds of doubt. So yesterday we discussed everything about the mitosis, right? So I have few questions. Let me see if you can answer or not. So mitosis, let's suppose that if I have a person or an organism which is having 2n as its chromosome, which means it is diploid, and if it is diploid after meiosis, what will be the number, after mitosis, what will be the number of chromosomes it is going to have? Will it be 2n? The option 1 says 2n, the option 2 says 4n, option 3 says n, and option 4 says none. So what can be your answer? Let me know in the comment section. So yes, very good. The answer will be what? The answer will be none other than but the 2n. Why, sir? Why? Because in mitosis, equational division takes place. In mitosis, what happens? Equational division takes place. Yes or no? Yes, sir. And that's why we will be having our answer as 2n. I hope that this is pretty much clear to you all. Now, before going to the meiosis, again, I'm going to make you revise the cell cycle. Okay, sir. So what was the cell cycle? Various steps of cell cycle we discussed yesterday, right? And those cell cycle, those steps will be pretty much similar in the meiosis also. So let us recall again. So what we had this cell cycle, and the cell cycle was divided into how many phases? It was divided into two phases. One was interphase and the other was M phase. Yes or no? Do you remember that? That's very good. So interphase was again divided into how many parts? It was G1, S and G2. Very good. So let's make here G1, S, and then we had G2. Clear? Now, after this, let me ask you a question. So the question says that in which of the following histone protein is synthesized? Come on, let me see some answers in the comment section. In which of the following the histone protein is formed? Okay, so the histone protein is formed in whether the option G1, S or G2. In which? We have the histone protein in the S phase. So histone protein is formed in the S phase. That's very good. Okay, now the only difference in the mitosis and the meiosis in the cell cycle is what? The M phase is divided into again two phases. Yes, the M phase is divided into two phases. So we have phase 1 and phase 2. That means prophase 1, metaphase 1, anaphase 1, telophase 1, and in the similar manner again it will repeat. How? Prophase 2, metaphase 2, anaphase 2 and telophase 2. So in mitosis and meiosis, this is the major difference. This was the M phase. Clear? Now, before moving forward, I like to tell you that this metaphase, like M phase 2 of the meiosis, is very much similar to the mitosis. So in this particular meiosis, we are only going to concentrate on the M phase 1, because that is where the main thing is happening, and M phase 2 is basically what it is very much similar to the mitosis. So are you ready? Are you ready to clear the concepts of your meiosis thing? If yes, give me some likes, and if you are new to this channel, subscribe to this channel, because we droppers are going to become toppers through this channel, through this series. So let's discuss about the meiosis. Okay, what is meiosis? If I ask you what are the characters of meiosis, so how many of you know about the characters of meiosis? Let me see some answers in the comment section. How many of you know the characters of meiosis? So here I am going to say some of the characters. The number one is what? Meiosis is used when? When do we use meiosis? If suppose you have got a cut in your hand, at that time you are going to use meiosis, or if you have to produce gametes. So sir, when we have to produce gametes, at that time we are going to do what? We are going to use meiosis. So that means what? Meiosis is basically used to form the gametes. Yes or no? Meiosis is used to form the gametes. So whenever gamete formation is required, we are going to use what? We are going to use meiosis. Okay. Now, meiosis is also known as what? It is also known as equational division, or what? No, meiosis is not an equational division. Why, sir? Because if you have a 2n cell, this 2n cell is going to get converted into n. That means a diploid cell is getting converted into an haploid cell. That means this is not an equational division like the mitosis. In this, the chromosome, the ploidy is decreasing. That means it is a reductional division. What is it? It is a reductional division. Now, reductional division. Why reductional division? You understood. Because here 2n, whatever the number of chromosome, it's suppose if it's 4n, after meiosis it will become 2n. If it is like 8n, after meiosis it is going to become 4n. Clear? So number of chromosomes will be reduced to half. That's very good. Now, this meiosis ensures that in a particular organism, let's suppose we are what? We are diploid, right? So in our life cycle, this meiosis ensures that haploid cells come, or haploid part come. So without meiosis, we can't imagine having the haploid cells, right? So I can write that ensure. What it ensures? It ensures haploid phase in our life cycle. Who are we? We are a diploid organism, right? Ensures haploid phase in diploid organisms. Now, if these things are clear to you, we will be moving forward, and if you have any kinds of doubt, let me know below the comment section. Now, let's move on and let us discuss the major part of our meiosis. Clear? Okay. So, this is what? This is some important points of meiosis. So, we discussed one thing. What's it? That here we are having 2 cycles of the M phase, right? So, this meiosis basically what? This meiosis is divided into 2 parts. What, sir? One is the interphase and the other one is the M phase, okay, which was pretty much similar from the mitosis. So we have interphase here, and second what? Second, it is divided into M phase. So let us write this M phase with some beautiful colors. So we have M phase here. Clear? Okay, sir. Now, the interphase, similar to the mitosis, very similar to the mitosis, it is divided into G1, S and G2 phase. Yes or no? Yes, it is. G1, S and G2 phase. And in the similar manner, if I talk about the M phase, here M phase is little bit different from the mitosis. The M phase is divided into M phase 1 and M phase 2. Clear? Now here M phase 1 is divided into what? It is divided into prophase, metaphase, anaphase and telophase. Okay, so if it is prophase of M phase 1, so we are going to say prophase 1, we are going to say metaphase 1, we are going to say anaphase 1, and similarly we are going to say telophase 1. But the same thing if it happens in the M phase 2, so we are going to write prophase 2, metaphase 2, anaphase 2 and telophase 2. And you know that this particular thing is very much similar to the mitosis. So we are not going to pay attention to this, rather we are going to pay attention to the meiosis 1. Now here in meiosis 1, the prophase is also further divided into lots and lots of stages. So we are going to discuss everything, but let's have some basic knowledge of each and every terminologies, and then we are directly going to jump into the cell cycle. Okay, now let's move on and let us understand there are term which is again what, which is homologous chromosome. Okay, let us understand that term again, although we discussed in the previous class, but let me revise if you are a new student here. So I don't want you to have that confusion that what was homologous and what was not. So let us have a quick recap of this homologous chromosomes. Okay, great. So see, if I am talking about the homologous chromosome, so did you remember that we had two guests in our previous lecture? One was Harry and one was Julie, right? So this is Harry and where is Julie? This one was Julie. So they created what? They created gametes. First of all, they both are human beings, and if they both are human beings, then what, sir? They will be what? They will be 2n, right? They will be what? They will be diploid, yes. So this will be 2n and this will be also 2n. Clear? This will be 2n and this will be 2n. Okay, sir? Right? Now, if they are going to produce what? If they are going to produce the gametes, so they had this type of chromosome before the production of gametes. They are having this type of chromosome, right? And again, they are having what? They are having this chromosome. Since they are diploid, so each chromosome will have one pair, each chromosome will have its pair, right? Okay, now they are going to form the gametes, and during the gamete formation, one will come from what? One will come from this Harry and the other will come from this Julie, right? And they formed the gametes like zygote, N and N, they mixed and formed the zygote. So this thing is known as what? This thing, these chromosomes are known as the homologous chromosome. This was the homologous chromosome. Okay, we discussed this thing in a very detail in the previous class. We have given lots and lots of time in this concept. Okay, now let us move on and let's see that if this chromosome underwent into S phase, so what was happening in S phase? In S phase there was the division, right? Duplication of DNA. So in S phase these chromosomes were getting duplicated. So how, sir? How? See, this was what? This was the normal chromosome, but when it entered into the S phase, it became what? Like this. Clear? And Julie's chromosome also became like this. Clear? So can you see the difference? This was before S phase and this was after S phase, and this will go into what? This will go into the G2 phase, and from the G2 it is going to go into the M phase. Clear? Okay, sir. Now these two chromosomes were known as what? These two chromosomes were known as the sister chromosomes, or sister chromatids, right? These two chromatids were known as sister chromatids, and everything everything we have discussed in the previous class, right? Now, next, what about these two, sirs? This two. So this two is known as what? Non-sister chromatids. This two is known as what? Non-sister chromatids. Okay, clear? Now, let's move on and let us understand the other terminologies. Okay, so I'm going to make a flowchart for you, and with the help of the flowchart, we are going to clear all the doubts. Clear? So this flowchart, I'm not going to focus much on G1 S G2 because we already discussed in the previous video. If you have not watched that, please go and watch that video after the class. That's a video full of concepts, full of contents, and full of those type of crystal clear concepts which are going to give you marks in your NEET examination. So basically they were discussed why 2C, why 4C, what I made you visualize like what is 2C and what is 4C. I can't explain in this video again because it will take lots and lots of time. So if you want to grab those concepts, you need to go back to that video. I'll ask the team to put the link of that video. Now, meiosis is divided into how many parts? Meiosis is divided into two parts, sir. Meiosis 1 and meiosis 2. I should say M phase of meiosis is divided into meiosis 1 and meiosis 2. That's great. Okay. If you are watching this video, do keep pen and copy with you so that you can write all the important points. Meiosis 1 and meiosis 2. Clear? Okay, sir. That's very much clear. Next what, sir? Next we are going to have this thing which is meiosis 1 and meiosis 2. We are going to divide meiosis 1 and meiosis 2 here. So meiosis 1 we are going to divide as prophase 1. The very first thing which comes here is prophase 1. So let's write here. Let me zoom in so that we can write all the things in this particular chart only. We have prophase 1. Okay, and now in prophase 1 we have some other stage also. What, sir? We have liptotene, we have zygotene, phakietine, diplotene, and diakinesis. So let me write here for you. We have this prophase 1. So prophase 1 is divided into 1, 2, 3, 4, 5 again some stages. So the very first one is what? The very first one we have is the liptotene. It is liptotene. And second one we have is zygotene. Second one, everything I'll make you remember in this class only, don't worry. Zygotene. And next we have phakietine. Next we have phakietine. And next we have diplotene. What we have? We have diplotene. And then we have diakinesis. What do we have? Diakinesis. Clear? Okay, sir. That's very clear. Liptotene, zygotene, phakietine, diplotene, and diakinesis. Again, let's revise it. Liptotene, what do we have? We have liptotene, we have zygotene, we have phakietine, we have diplotene, and we have this diakinesis. Okay, let's revise it again. Liptotene, zygotene, phakietine, diplotene, and diakinesis. Clear? Now, what happens in this thing? Here, we are going to have maximum questions. Majority of the questions are gonna come from this part. So, stay concentrated. This liptotene and zygotene is basically what? They're short-lived. They are not very long. They are basically what? They are short-lived. So, let's write here. These stages are short-lived. Okay, sir? They are short-lived. We understood that they are short-lived. Now what, sir? Now the thing is that what happens in these stages? So in liptotene, chromosomes gets visible. Remember, it's a stage of prophase. So in prophase of mitosis, what happened? In phase of mitosis, it was what? It was the phase where the chromosome was getting what? They are getting condensed. So in the similar manner, the very first step of this prophase 1 of meiosis 1 will be what? In liptoteen, the chromosome will become visible. It is going to get condensed. So I should write here, in liptoteen, chromosomes gets visible. What happens here, sir? Chromosomes gets visible. Okay, we're going to write chromosomes gets visible. Now what, sir? Now we have zygotine. What we have? Zygotine. After liptoteen, we have what? We have zygotine. So in zygotine, what happens? There are some terminologies which is I'm gonna say you, and we are going to discuss each and every terminology. We have synapses, we have synaptonymal complex, bivalent, tetrad, everything. All these terms, whatever the question comes here, it is happening under this zygotine. So what terms I used? I used synapses. I used synapses and used synaptonymal complex. Sir, what are these? I will going to discuss it in a very short period of time. So let's make a flow chart so that you will remember everything here only. You don't have to read 5 to 6 pages of your notes. Just focus on this. Let's make this and we are going to discuss everything. Synapses, synaptonymal complex, and what? Bivalent, and what? Or tetrad. Or tetrad. So these are what? These are the terms which we are often hearing and which is making us confused, right? So today will be the last day of your confusion because everything we are going to discuss here. Now let's come here, let's come to the next stage. And what is the next stage? We have fecitating. The next stage we have is what? We have is fecitating. So what happens in fecitating, sir? In fecitating, major thing of meiosis occurs. Any guesses? Any guesses? What is the major thing of meiosis? Why meiosis is happening? To exchange the genetic genes of some of the chromosomes, right? So, and that is what? That is basically crossing over. So mainly in fecitating, what happens? Crossing over takes place. Crossing over takes place. We sometimes what do now? We sometimes directly jump to the concept. Okay, crossing over, we will see what is crossing over, but we are not, we are just like, we don't know what is crossing over, what's happening, at least when we make a mind map, and then we are going to study the concept. See, your concept, you are going to able to visualize the concept. Okay, that first of all chromosome was able to get visible, and then bivalent formed and crossing over is happening. So crossing over takes place. Now what is crossing over? We don't know right now, but we are going to see it. Next we have diplotin. So diplotin, what happens? Whatever this complex were formed, that is synaptonymal complex or synapses or bivalent or tetrad, everything gets what? It gets dissolved. So I'm just gonna write here that synaptonymal complex gets dissolved. Okay, and there is one more thing here which is very frequently asked in NEET. It's one of the favorite question of the NEET examiner, which is what? Which is chiasmata. They are going to ask you that where the chiasmata is formed. So the chiasmata is formed in the prophase 1 of diplotin stage of prophase 1, right? So let's write here chiasmata. I'm gonna make it bold. Chiasmata. This is a very hot area from where the question can come. Star mark it. Lots and lots of questions we have come. I'll show you. We are going to have a detailed quiz on Sunday at 11 o'clock for this particular chapter, and I will be bringing lots and lots of questions. And my question making criteria is very different from others. I myself solve the questions, and if suppose in any other questions I am taking more than 30 seconds or 20 seconds, that means that questions can be a little tricky for you, and I am going to put those questions on the priority, whether it's PYQs or it's from n number of books. So you don't need to get worried about the quality of the questions. We are going to have a very top-notch questions on Sunday at 11 a.m. Now let's come back to the dikinesis. So what is dikinesis? What happens in dikinesis? Terminalization of chiasmata. Just remember, chiasmata was formed where? Chiasmata was formed in the diplotin, and where it got terminated? It got terminated in diakinesis. It got terminated where? It got terminated in diakinesis. So now I think so it's clearly visible to you. So what is this? Terminalization of chiasmata. Terminalization of chiasmata. Yes, okay, clear. Now what happens here? See, this is the end of prophase 1, right? This step is what? This is the end of the prophase 1. So what happened in mitosis of prophase? So in mitosis, during the end of the prophase 1, we got to know that the nuclear member disappeared, cell organelles got disappeared, these things happened. So yes, everything is gonna happen here also. Nuclear membrane will get disappear. Okay. Now what? Cell organelle will get disappear. Okay. Nuclear membrane, cell organelle, all these things will get disappeared, right? And nuclear envelope also, we have written that, right? And what is gonna happen? The cell is now going to get ready to the metaphase, right? So here what's happening, I can write here, transitioning is happening. Transition to the metaphase is happening in what? In diakinesis. So let's write here, transition to metaphase. Transition to metaphase is happening. I hope that this much is clear to each and every one of you. I'm going to be little side so that you can take the screenshot of this prophase 1. So again, let's revise it quickly and let's understand about the prophase 1. Okay. So meiosis, M phase of meiosis is divided into two parts, meiosis 1 and meiosis 2. In meiosis 1, we have prophase 1, metaphase 1, anaphase 1, telophase 1, and right. And then in prophase 1, what is going to happen? We have liptotin, we have zygotin, we have fecitine, we have diplotin and dikinesis. What are the major things happening in the liptotin? So sir, in liptotin, chromosomes gets visible. What is happening in zygotin? In zygotin, synapses is formed and synaptonemal complex, bivalent or tetrad is formed. What is happening in fecitine, sir? In fecitine, crossing over is taking place. And what happening in diplotin? All those complex which were made, that's got disappeared and new structure was formed, which is chiasmata. And in dikinesis, what's happening? That chiasmata is also gone and nuclear membrane gone, nuclear envelope gone, cell organelles gone, everything disappeared, and the cell is now being transitioned to the metaphase 1. I hope that this thing is pretty much clear to each and every one of you. Now, if this is clear, let's move on forward and let us now understand about few more things. What few more things, sir? We are going to understand about the terms. Whatever we discussed, synaptonemal complex, synapses. You don't want me to discuss or what? Should I just go make the flowchart and go from here? No, I am going to discuss each and everything, don't worry. So, let us talk about the liptotin first. Let's talk about the liptotin. Okay, liptotin. This cell chapter is very easy, if you get to understand the concept, it's very easy. When I was in class 11, the very first time I entered my school with biology subject, and I was like, oh my god, the bio will be very easy because class 9th and 10th bio was nothing. We just have to read human reproduction. That was also very short topic, and then we had some neural control and coordinations and all those things. And in class 9th, it was why do we fall ill, animal kingdom, plant kingdom, and I was like, oh, okay, that is very easy. But the very first time when I entered into my school, the teacher was like, let's start with mitosis and meiosis. And I was like, okay, here starts the game. So now, but I was not able to understand anything at that time, but I think so you are able to understand all of these things. So if this is, you are able to understand this particular thing, then most of the things in biology you are able to understand, because this is a very great concept. And if this is clear, your biology will be very easy for you. Now, let's get back to the topic. We have what? We have liptotin here. Okay, so we have the liptotin. What happens in liptotin, sir? In liptotin, the chromosomes are what? The chromosomes are loosely condensed, okay? Like it's just the starting. Chromosomes are just starting to condense. So what I should write here? They are loosely condensed. Okay, sir? We understood. They are loosely condensed. Loosely condensed. Okay, now sir, what are the other properties? Other properties we have what? We have here longest and the thinnest chromosome. In this particular phase, what you're going to have, you're going to see that among all the phases, if you're going to see this phase under the microscope, your chromosomes will look like they are very big and very thin. So I'm going to write here longest and thinnest chromosomes are found in liptotin. Very good. Longest and thinnest. Keep an eye on me. I make lots and lots of spelling mistakes. Okay. Longest and thinnest chromosomes are found in liptotin. Very good. And this stage is known as what? This stage is known as Bouquet stage. This stage is known as what? Bouquet stage. Okay. Question can arise. They can give you the name of the stage, or what they can do, they can just ask you that tell me where the longest and thinnest chromosome. So liptotin. Clear? This was all about the liptotin. Now I'll make a diagram for you, a beautiful diagram for you. This is a cell, okay, and inside the cell what do we have? Nucleus. And somehow the chromosomes will be very simple, like very simple. It will be just like this and this. Longest and the thinnest chromosomes are found in liptotin. Is it clear to everyone? If it is clear, like this session, subscribe to this channel and share this session among your friends. Now let us move on and let us discuss about some of the other things like zygote. This is the main thing of the meiosis. If you are going to understand this thing, your meiosis is clear. Yeah, I'm not joking. It's very important. The major thing is happening here. In zygote and fecitine, all the things are happening here. Okay. Now, zygote is also known as synaptotene. What's the another name for zygote? The another name for zygote is synaptotene. See, the another name are also very important. I remember there was one of my student who just passed 12th and he started to study for NEET. And somehow it was like he didn't got much time to study for NEET after the boards. So there was, he pretty much know what are sexually transmitted disease, right? And Khadija ma'am is taking your zoology section. So when she is going to cover the reproductive health, she will definitely tell you. But in exam, what happened? The question came about venereal disease, right? And those who know, those who have read this chapter, they both had the same thing. But this particular person was like, sir, I don't know from where this question came. And I showed him the line. See, it's given. So that's why we shouldn't ignore the second word. It's like, okay, I'll remember the first word, but if it comes in exam, your mind, it will slip out from your mind and you will forget. So do focus on the second word also. It is known as what? Synaptotene. Synaptotene. Okay, that's great. Now let us make a beautiful diagram for this particular thing and leave everything what you watch on the screen. Your concentration should be on your screen. Okay, now this was the homologous chromosome. You remembered Harry and Julie? Yes sir, Harry and Julie. Let's have Harry's chromosome. Yeah, was this a homologous chromosome? No, this is not a homologous chromosome. This is a chromosome from S phase, the duplicated chromatids. How this is the homologous chromosome? When there will be what? There will be Julie's chromosome, then it will become homologous chromosome, right? So this is the Julie's chromosome. Now both are what? Both are the chromosome number 1. Like from Harry also we have taken chromosome number 1, and from Julie also we have taken chromosome number 2. Sorry, chromosome number 1, what I am saying. Again, this from Harry we have taken chromosome number 1, and from Julie we have taken chromosome number 1, and this is how they make the homologous chromosome, right? Yes. Now these homologous chromosomes are going to do what? They are going to form a complex which is known as synaptonymal complex. Okay, sir. What is going to happen here, sir? See, the pair is basically what? They are going to have some depositions, some proteins here, and this is going to known as synaptonymal complex. Okay, so we understood that this is going to be the synaptonymal complex. Synaptonymal complex. Very good. We got to know. But sir, you have told so many terms. Bivalent, tetrad, synapses. What are those? See. So actually the pair, these are the pairs, right? How many chromosomes we have? We have 2 chromosomes here. One from Harry, one from Julie. So Harry and Julie was what? They were parents, and this, the chromosome is of a child of Harry and Julie. So these are the homologous chromosome, and when they are making the pair, we call the pair as synapses. We call the pair as what? We call the pair as synapses. Okay, what they have formed? They have formed synapses. Is it visible to you? They have formed synapses. Clear? Okay. Now, sir, what happened, sir? So zygote, actually what happened in zygote? How the synaptonymal complex is formed? Basically nothing happened here. Pairing of homologous chromosome, right or no? See what we have done. We have just paired the homologous chromosome. So pairing of homologous chromosome happens where? Happens in the zygote, and this is what? This is the formation of a synaptonymal complex. Okay, homologous chromosome. Pairing of homologous chromosome happens where? In zygote. Okay. Now what happens? What happens, sir? Now see, we are going to discuss about few more terms like tetrad and what? And bivalent. Okay. When there is a pairing of homologous chromosome, the complex which is formed is what? The complex which is formed is the zygote. Sorry, synaptonymal complex. Okay, synaptonymal complex gets formed. Clear? Now, the pair is known as what? The pair is known as synapses. Clear? Now we have 2 terms, tetrad and bivalent. Why tetrad, sir? Why tetrad? Why we are calling these structures as tetrad? So can you see here, if I tell you to watch this thing, this is what? This is one homologous chromosome, and this is what? This is the second homologous chromosome, right? This was the Harry's chromosome, and this was the Julie's chromosome. Both for what? Both for the chromosome number 1. Clear? So they are homologous chromosome, and they are what? They are pairs, right? So how many pairs do we have? How many pairs do we have? We have only one pair. So this is known as synapse. Clear? Synapse clear. Now, how many chromatids we are having here? Total number of chromatids. How many chromatids we have? So can you count it? This is having 2. This is having 2. So 2 and 2 makes what? 2 and 2 makes 4. So how many chromatids we are having? 4 chromatids, and that's why we are using the term tetrad here. 4 tetrad. Clear? And now, why bivalent, sir? Why bivalent? So I'm gonna ask you again. This was what? This was one homologous chromosome. This was 2 homologous chromosome. This was 1 chromosome and this was 1 chromosome. Okay. How many chromosomes do we have? We had 2 chromosomes, and that's why it is known as bivalent. Again, I'm gonna repeat it. Why tetrad, sir? We had 1, 2, 3, 4 chromatids. That's why tetrad. Why bivalent, sir? We have 1 chromosome, this one, and 1 chromosome, this one, which is what? That's why bivalent. So bivalent term is used in reference with chromatids. Bivalent term is used in the reference with, chromosomes. With reference with chromosomes. How many chromosomes, sir? 2 chromosomes. That's why we are telling it as bivalent. Okay. And tetrad term is used with respect to the chromatids. How many chromatids? 4. That's why tetrad. Now I hope so that this particular thing is clear to you. What is synapse? The synapse is the pair of homologous chromosome is synapses. What is synaptonymal complex? When they paired with each other, that is known as the synaptonymal complex. Clear? Okay, sir, that's very much clear. Now let us move on, and let us move on with the another stage, which is what? Which is the fecitine. Clear? Which is the fecitine. So after, obviously, we had prophase 1. Prophase 1, may what we have discussed? We have discussed the leptotene, we have discussed the zygotine. Now what comes? Now comes the fecitine. Now comes the fecitine. So let's write here fecitine. Fecitine. Okay, I'll do this thing little down so that it's visible to you. Let's have the fecitine. Okay. Now in fecitine, again, this is also a major part of the meiosis. This is also the major part of meiosis. And in meiosis, everything, whatever the important concepts which we have, it is only and only in the prophase 1 only. After that, it's nothing. It's very simple, right? So we have fecitine. So in fecitine, what happens? Crossing over takes place. In fecitine, what happens? Crossing over takes place. Sir, what is crossing over? So see here. What happens here is that, let's call Harry again. So this is the Harry's chromosome. And who were Harry and Julie? For that, you need to watch our previous sessions, right? So now, this was the Harry chromosomes and Julie was pink in color. So I am gonna take this Julie as pink in color, okay? So they were what? They were the parents, right? Harry and Julie were the parents. And this chromosome is of what? This chromosome is of their child. Now, what happens? So we are going to do the crossing over. Yes, definitely. Now here, can you see the tetrad? This structure is tetrad only, na? The whole structure, can I say it as a tetrad? Yes, definitely, sir. You can say, because this is a tetrad. We can easily see 4 chromatids here. How many chromatids? See this one chromatid, this one chromatid, this one chromatid, and this one chromatid. My finger, yeah, 4 chromatids. Clear? We have this 4 chromatids. So 4 chromatids means tetrad. Now what happens here is that they are going to do some mix and match. They are going to perform crossing over. So see here, some part my chromosome became lot thicker, so let me make it little nicer. Okay, this was the Harry's chromosome and this was the Julie's chromosome. So they are going to do what? They are going to exchange their genetic material. They are going to do what? They are going to exchange their genetic material, and this is only known as crossing over. And you were like, sir, crossing over, मेरे को नहीं समझ में आता, I don't know crossing over, sir. What do we do? This is crossing over, just so simple. So what is going to happen here? Now let me do one thing for you. I'll make this particular chromosome little darker. It's looking like both are white. It gives me a feeling that I am making some flowers, beautiful flowers. I'm a very good artist. As you can see, my drawings are very good. No one can make better drawings than me. Yeah. Okay, now let's move on. So when this crossing over will takes place, it will be finished. So this is how we are going to see the chromosomes. Okay, so half of the portion, or half or some of the portion of Julie's chromosome will be in Harry's chromosome. Yeah, and half of the portion of Harry's chromosomes will be in Julie's chromosome. So this is what, this is the crossing over. And if I ask you one thing, Okay, what happened to my screen? Just a second, I will reshare my screen again. Just a second. A small disturbance in my class again. Okay, guys, just a second. Okay, so we will be back soon. Let us discuss about the Harry's chromosomes and the Julie's chromosomes. Okay, so what we were discussing about the crossing over. And if I ask you the question about the crossing over, like how and what is crossing over, so this is the point where things is gonna change. How, sir? How things is gonna change? We always says that meiosis is responsible. We always says that meiosis is what? Meiosis is responsible for the generation of variation. Yes or no? We always discussed about this. And this is the reason for what? This is the reason for the variations, guys. See, the genetic materials are what? The genetic materials are getting changed, they are getting mixed, and that's how we can say that crossing over is taking place. Yes or no? So this is the reason where the variations due to the sexual reproduction is produced. I hope that this one thing is clear to you. And I think so that I am back with my screen. Yes or no? Yes. That's very good. So just give me a second. I will be again back with my screen. Okay, what happened to my screen? Just a second, guys, just a second. Yes, I am back with my screen. Okay, understood everything? So let's discuss again. We had what? We had this tetrad we had. Okay, this tetrad having what was tetrad? We were having 4 of the chromosomes, 4 of the chromatids, right? So 1, 2, 3, 4 chromatids were there, and then they did what? They did the crossing over. See here, the crossing over started. Okay, and when the crossing over ended, what happened is that the Julie's chromosome was having what? Were having some of the part of the Harry's chromosome, and Harry's chromosome were having some of the part of the Julie's chromosome. So I can say that here what happened? The crossing over took place. What happened, sir? The crossing over took place here. Clear? Okay. Now let's move on and let us discuss about some of the other terminologies which is very important for us. Now, see. So in zygote, everything is happening what? Everything was like zygote tetrad we have saw, and then it came where it came to fecitine, right? So in fecitine, what is gonna happen? In fecitine, crossing over is taking place, and during this thing, during the fecitine, there is an enzyme which is needed very much. Can you tell me about the name of that enzyme? What is the name of that enzyme? Anyone? Anyone in the comment section? There is an enzyme which is needed during this particular fecitine, during this particular crossing over. Anyone? I can see some of them, you are live. Let me know about the answer. Let me know about the answer. If you have any kinds, if you have any hint, should I give you a hint? Okay, let me give you a hint. The hint is basically what? It starts with the name R. It starts with the name R. Which enzyme? I repeat my question. Which enzyme is responsible? Which enzyme is responsible for the recombination or for the crossing over? I gave you a hint. I'll give you a hint right now. So the enzyme which is involved in this particular thing, which is the crossing over, is nothing but the recombinase. The enzyme which is used here is recombinase. So let me write here, what is the enzyme name? Recombinase. Clear? So everything, whatever the thing is happening in this particular stage of fecitine, it is just because of what? It's just because of this particular enzyme. Clear to everyone? That's great. If you have any kinds of doubt, let me know in the comment section. Okay, let me know in the comment section. Just a second, people. Okay. I have, yes, we got back. Okay, so that enzyme which is used here will be what? It will be recombinase enzyme. Which enzyme? Recombinase enzyme. We are going to use the recombinase enzyme. Okay, now what else is happening in fecitine? Let us understand everything, because questions can come from these points. The questions are these points are very hot topics. In meiosis, if questions is gonna come, it would be crossing over. It could be about the fecitine. It would be about tetrad bivalent. They are very hot topic. So in fecitine, again let us write what are the things we saw. We saw that crossing over is taking place in fecitine. Yes, sir. Tetrad is very much clearly visible here. Here, tetrad is clearly visible. Tetrad is clearly visible here. Okay. Yes, sir. And then what? Recombination nodules is formed. What nodules? See, this we have seen here. This part is known as what? This part is known as recombination nodule. What is this, sir? Suppose this is one chromatid, this is the other chromatid, and this structure is what? Recombination nodule. So I can write here, recombination nodule is formed. Formation of recombination nodule is there. Clear? Yes or no? Yes, sir. It is pretty much clear. Now what, sir? Now we have the enzyme. What enzyme we discussed right now? Yes, everyone, fast. What was the name of that enzyme? The name of that enzyme was, sir, it was recombinase. It was recombinase enzyme. And this recombinase enzyme is basically what? This recombinase enzyme is basically endonuclease. That means it is gonna cut the DNA from these parts. Like suppose that this is the, I'm going to make you understand little bit what is endonuclease. So let's suppose this is a chain of something, a chain of molecule. Let us understand this only. This is a chain of molecule, and this endonuclease, the recombinase enzyme is what? It is a category of enzyme which is known as endonuclease. So it is gonna cut these things like this from the endonuclease, means from the inside it is gonna cut them. Okay, these kind of enzymes are known as endonuclease enzyme. So what kind of enzyme do we have as recombinase? So recombinase is an endonuclease enzyme. Although we are going to discuss these endonuclease, exonuclease, all these enzymes part in the biotechnology. That will be the where you are going to get a correct idea that what actually these are. Or we are going to also discuss some of the part in the enzyme section of class 11th. Clear? Now, right now you just have to remember that this enzyme is what? It is an endonuclease enzyme. Okay, crossing over finished, fecitine finished. Okay, if crossing over is finished, then Fecitatin is finished. Let's write crossing over also. Although I have written here, but let us mention it again. Clear? So this was the crossing over. After this, what we have? We have diplotin. After fecitatin, what we have? We have diplotin. And now everything is pretty much easy. Very easy. Seriously, everything is now very much easy. If I am going to talk about diplotin, so in diplotin, there will be the formation of chiasmata. There will be the formation of chiasmata. So whatever the synaptonymal complex were formed, it will dissolve. First of all, the complex which was formed here, see, the complex which were formed here, let's have this complex. Let me do a little cheating by copying this diagram to here. Okay, I hope that you haven't seen anything. Now let's see. Let us erase this complex, synaptonymal complex. Okay, just the synaptonymal complex I need to erase. Yeah, so this synaptonymal complex got dissolved. Okay, synaptonymal complex dissolved. So let's write here synaptonymal complex dissolved. Okay, sir. We understood that the synaptonymal complex got dissolved. Now what? Now there will be the formation of chiasmata. Now there will be the formation of chiasmata. How, sir? So let's suppose this is like this, and this was the Harry's chromosome. Okay. This was the Harry's chromosome and this was the Julie's chromosome. Okay. See. So how this will be formed? They are going to make this kind of structure, and this is known as what? This is known as chiasmata. This point will be known as what? Chiasmata. Okay, chiasmata form. It's just like we tie a little a thread. We tie a thread and sometimes it develops a knot. So similarly, chiasmata is kind of a knot, and slowly, if suppose we have a thread and we do it like this, so that knot moves upwards, right? So here also the chiasmata slowly it is going to be removed, but it is not going to be removed in diplotin. It is going to be removed in the later stage, which is dikinesis. Now, in human reproduction, we always says that sperm cells are made throughout the life cycle of a male, but the egg cell is made inside during the embryonic life only. Whatever the oocyte is required, they are made, right? And here the oocyte is basically what? That oocyte is stopped in they are paused in the diplotin stage only. So oocyte is basically what? It's a very good question. A question can come that oocyte is what? Oocyte is paused in the diplotin stage, Clear? I hope that this is clear to everyone. Diplotin clear? Yes, sir. Next, let's go towards the dikinesis. Let us talk about the another thing, that is dikinesis. What is this, sir? This is the most easiest thing. What is going to happen? Terminalization of chiasmata, right? So here we are going to make a full blown diagram. So what was the Harry's chromosomes now? The Harry chromosome after the crossing over was like this, right? Half here, half here. And what was about the Julie's chromosome? So Julie's chromosome were also like this after the crossing over. Okay, and they had formed the chiasmata. So chiasmata was something like this. And after this, what happened? One second. See, they were like this. I'll copy them first of all, and I'll make you understand that what's actually happening. See. Okay. So during the diplotin, they formed a chiasmata. Okay. During the diplotin, they formed the chiasmata, just like this structure. Okay. Here also you can mention that half of the thing is of Harry's is in Julie, and half of the Julie's chromosome is in Harry's chromosomes. Okay, and they form the chiasmata. Now this is what they are looking right now. In dikinesis, they both are going to be separated. That means chiasmata is dissolved. Chiasmata is terminalized. So terminalization of chiasmata. Terminalization of chiasmata means that this chiasmata which was formed, slowly it will go to the end and it will terminalize, it will dissolve. So terminalization of chiasmata takes place where? Takes place in the diakinesis. I hope that this thing is clear to everyone. Terminalization of chiasmata takes place where? In dikinesis. Now what? This stage, as we discussed, represents the transition to metaphase. Transition to metaphase. And this we have completed the prophase 1. Let's quickly see the flowchart which we have made and let's revise it very quickly. Okay, so we have discussed about this thing. Prophase 1, leptotin chromosome gets visible. That's clear, that we have discussed. In zygote, synapses, synaptonemal complex and bivalent and tetrad is formed. So everything now we know. What is tetrad, what is bivalent, we all know. Now we found out what we discussed about the fecitine crossing over, we discussed. Okay, and in diplotin, synaptonemal complex was getting dissolved and chiasmata was getting formed. Okay, and after that what happened? Chiasmata got terminalized in dikinesis, nuclear membrane got disappeared, and then this was the phase where the prophase is ending and the cell was transitioning to the metaphase 1. Clear? Now, after this prophase 1, what comes after this prophase 1? We know that metaphase is gonna come. So let's write about metaphase now. Okay, let us write some point about the metaphase and then we are going to discuss it again. So in metaphase, nothing special is going to have. What was happening in the metaphase of mitosis? In metaphase of mitosis, sir, all the chromosomes were aligned to the equatorial plate. Yes, that's only. So here we are going to say that bivalent. Again, sir, you are using a term bivalent. Why can't I use the term bivalent? Because the pair which we have, the pair is what? The bivalent, right? Bivalent refers to what? The pair of that homologous chromosomes, right? So we, in that pair of homologous chromosomes, how many chromosomes were there? 2 chromosomes. So I can say that bivalent, right? We haven't said in prophase 1 that bivalent chromosomes, that chromosomes are not separated yet. They are still with them, right? They are in pair. So I'm going to write here bivalent chromosome. So I can write this term. I can use this term right now also. Bivalent chromosome, right? But after this anaphase 1, I can't use this term bivalent. Okay, I will discuss that. So bivalent chromosome, where it is going, aligns themselves in the metaphasic plate or equatorial position. Bivalent chromosomes aligns at equatorial plate, yes or no? Or just write equator. Clear? Okay, sir, we got it. And then what is happening? Spindle fiber, you remember in metaphase of mitosis, spindle fiber generated by the centromeres, they came and they joined in the kinetochore and they pulled apart each other in anaphase, right? So similar thing is happening here also. So kinetochore gets attached, kinetochore, I should write like this: spindle fiber gets attached with kinetochore. Spindle fiber gets attached where, sir? In the kinetochore. In kinetochore. I'll make you understand again. Let's write the things and then again let's get to understand. So we have discussed about the major things happening in the metaphase 1. Now let us complete the metaphase 1 thing here. So let me have this metaphase. Let me draw the diagram for the metaphase 1, sir. Okay. So in metaphase 1, we discussed that chromosomes are what? Chromosomes are going to aligned in the equatorial plate. And what type of chromosomes? Bivalent chromosomes. What type of chromosomes? Homologous chromosomes, right? Okay. So let's suppose that this is a cell This is our cell. Should I draw a nuclear membrane here? Just let me know. Should I draw a nuclear membrane in metaphase 1? Yes or no? No. Why I can't draw? Because now the nuclear membrane is gone. Nuclear membrane is what? It is gone, right? Yes. So let me draw the chromosomes directly. We will have the chromosomes directly here. So the chromosomes will be kind of aligned like this. This was the Harry's chromosomes containing what? Containing the parts of Julie chromosomes, right? And again, this was something like this. Crossing over had taken place. And this was what? This was the Julie's chromosome containing some part of the Harry's chromosome. Okay. So let me draw here. And don't think so that in this arms crossing over didn't happen. It happened. We didn't draw it, but it will happen here also. So to avoid your confusion, let me draw the tuk white drops and pink drops here also. Okay. So this is the what? This is the bivalent. So all those bivalent is gonna get arranged in the equatorial plate, right? And then what happened? The centrosomes is going to produce a spindle fiber, and these spindle fiber is basically attached to the kinetochore. Okay, so this is the what? This is the structure of the metaphase 1. This is what is happening in the metaphase 1. Okay, what is this? These are the spindle fiber. These are the spindle fiber. Clear to everyone? Spindle fiber. I hope metaphase is very much clear. It's just like this. It's nothing more. Very easy. Okay. So we discussed spindle fiber is getting attached into the kinetochore. What was kinetochore, sir? We have forgotten. If you have forgotten, watch the lecture again, the previous lecture. But okay, I'm going to make you understand. So here is what? Here is the chromosomes, right? And this was the what? This structure was the kinetochore. Clear? This structure was the kinetochore. It was a disc-like shape where the spindle fiber comes and get attached. Okay. Now metaphase 1 is over, sir. Yes, metaphase 1 is over. Next what? Next let us go to the anaphase 1, and here comes the change. Majority of the thing is gonna change here. Pay very attentive, be very attentive, pay lots and lots of attention here, because majority of the thing is gonna change here. Okay, that's great. So in anaphase 1, what is going to happen? Homologous chromosome is gonna separate. Yes, what is gonna happen? Homologous chromosome separation is going to happen. So let me write here. Where should I write? I'll write here only. Okay, anaphase 1. Anaphase 1. What is going to happen, sir? Homologous chromosome separated. Homologous chromosomes separated. See, can you see this? Homologous chromosomes separated. How, sir? What does this mean? Let me show you the diagram. And this is the best part. If you understand this, your cell cycle is complete. Your cell cycle will be complete. See, right now, till now, how chromosomes were arranged? The chromosomes were something like this. These were what? These were the homologous chromosome, right? And during the metaphase 1, these chromosomes are what? These chromosomes are attached with the spindle fiber. Yes or no? Like this, like this. Now they are going to what? The spindle fiber is going to tear them apart, and these chromosomes will be like this. They will be what? Julie will be here. Julie's chromosome will be here. Julie's chromosome will be here with that crossing over thing. Okay. And then what is gonna happen here? What is gonna happen? Harry's chromosome will be in the other pole. So this is what homologous chromosomes for the very first time got separated in anaphase 1. Homologous chromosomes got separated in anaphase 1. Is it clear now? Now, sir, what is happening here is that just remember these were what? If this type of chromosomes were there, homologous chromosome were there, we were saying that it is a 46 chromosome. Yes? Yes or no? Right now, we were saying 46, 46, 46, 46. Now, what we have done? In those homologous chromosomes, we have separated the pair. That means this was N, okay? This was N, and they both were making what? They both were making 2N. This N, and this N. This was N, this was N. Both N were making 2N. Yes or no? Yes. Now what we did? We separated N and N, and this is what? The reductional division. At this point of time, the homologous, the diploid organism is getting converted into the haploid organism. The homologous pairs of chromosomes are separated. This is where the essence of meiosis lies. So this is how anaphase 1 is the main part of the meiosis, because here only the reductional division is taking place. Okay. So I can write here that what happened here? Reduction happened. What happened, sir? Reduction happened. The organism which was diploid is now converted into haploid. But do remember, these are not separated. This thing is still not separated. This will separate into anaphase 2. Clear? So I will write here that homologous chromosomes in anaphase 1, what happened? Homologous chromosome got separated. Homologous chromosomes got separated. Yes, sir. Very good. Got separated. But sir, what about the sister chromatids? What about the sister chromatids? So sister chromatids are still intact. Sister chromatids are what? Sister chromatids are still intact. They are not separated. See, they are not separated. These are the sister chromatids. They are not separated. They are still there. They are still there. Sister chromatids. Clear? Okay, sir. Now what is happening? Now after this, the cell will go to the telophase stage. After this, the cell is going to go into the telophase stage, which is telophase 1. So in telophase 1, what is going to happen? See, if I draw the whole cell, so what's going to happen here? Just give me a second. I'll explain you the thing. See here. What is happening now, sir? In anaphase 1, the cell was something like this, okay? The furrow formation got started, little furrow formation got started, okay, sir? And we had this chromosome, it got separated. Earlier they were what? They were homologous chromosomes, and now they got separated, okay? So, and now what is going to happen? They are going to divide slowly and make the two daughter cells, okay? So in telophase, the cell is going to be divided completely, okay? This was the anaphase. In anaphase, what happened? The chromosomes, homologous chromosomes got separated, and this was the telophase. So after the telophase, karyokinesis got completed along with the cytokinesis, okay? So here nuclear membrane will reappear in telophase, cell organelles is gonna repair, everything is going to happen, and at the end of the meiosis 1, we will be having two cells, right? Two daughter cells. Clear? Okay, sir. That's very good. So telophase 1, how can I write here? Telophase 1, we are going to write that nuclear membrane reappears, everything reappears, and reverse of prophase 1. Yes, of course, nuclear membrane appears, right? And whatever the things happened, condensation of chromosomes and all, they are again going to be reversed. And in this, what happens? Cytokinesis also takes place. cytokinesis is done. Now, what is this? What is formed? So, basically, here we had 2n. In the starting of the meiosis 1, we had 2n cell, right? Now, this 2n is what it is converted into 2 daughter cells with n and n chromosomes, half of the number of chromosomes, and we say them as dyad of cells. We say is dyad of cells. Is it clear to everyone? That's very good. Now the cell is like, I need a little rest. Meiosis 1 complete. After this, the cell is going to enter into the meiosis 2. Okay. So this cell will undergo meiosis 2. This cell is going to undergo meiosis 2. And again, it is going to form 2 cells. Again, it is going to form 2 cells. So at the end of the meiosis, we are going to have in total of 4 cells, right? And this you remembered when you might be studying about the sperm cells or anything. We got to know that sperm cells is a gamete and it is formed with the help of meiosis. So from one sperm mother cells, we are getting 4 sperms, and this is the reason behind that thing, right? So everyone will be what? N, 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 and N. Meiosis 1, this was meiosis 1. Meiosis 1, at the end of meiosis 1, 2 daughter cells. At the end of the meiosis 2, each daughter cell is going to give what? Each daughter cell is going to give 2 daughter cells. So in total, what we are going to get? In total, we are going to get these 4 cells. Clear to everyone? Yes, sir. It is clear. That's very good. Okay. Now, what do we have? We have the interkinesis. There is a term known as interkinesis. You might have gone to movie, obviously, everyone. So they give you a break like intermission, right? So here we have interkinesis. Interkinesis is what? Gap between meiosis 1 and meiosis 2. Interkinesis is what? It's a gap between meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2, right? This is the gap between meiosis 1 and meiosis 2. Okay. Now, sir, what? Now, the chapter is almost completed. Why, sir? Because meiosis 2 is what? It is similar to the mitosis. In meiosis 2, what we are going to see here? See, in the meiosis 2, we have again prophase 2. Now, don't worry. Here, the prophase is not divided into liptotin, zygotin, diplotin. Okay. Just we have prophase. So we are going to write here prophase 2. We are going to write here prophase 2. And this prophase 2. Now, I am going to ask you that how much chromosomes or how many chromosomes do we have in this particular thing? Whether the condition of cell is N or 2C, whether the condition of the cell is 4N, 4C, what is the condition? So, see here. Right now, before entering to the prophase 2, the cell is something like this, okay? Julie is what? Julie is like this, right? With some of the variations. Oh, white one was Harry. So Harry was like this with some variation, some genetic material of Julie. Okay, sir. Now, are you able to see 2 different chromatids? Are you able to see 2 different chromatids? Yes, sir. We are able to see one. But are these chromatids attached to the same centromere or different centromere? No, sir. They are attached with the same centromere. So this is what? This condition is basically N. We are going to say that this is basically what? This is N. Yes, sir. This is N. How many content of DNA? So this we considered as 1 content C. This considered as 1 content C. So this is what? This is the 2C. Yes or no? If you don't understand about the 2C, 3C, 4C, 5C, 6C, whatever, go back to the video of mitosis 1. There we gave a lot of time in understanding this thing. Okay. So go back to that video after this class and watch that. So we have this 2C. So each of the cell entering the meiosis 2 will have 2C content. Okay. Now prophase 2 done. Prophase 2, what is going to happen? Again, the same thing. Nuclear membrane gonna disappear. The our cell organelles is gonna disappear. Same thing like mitosis. Everything is gonna same. Okay, so don't worry about that. Let's see the major things. In prophase 1, 2, we have seen that condition will be like this. In metaphase, what was happening in mitosis? Say me quickly. Sir, in metaphase, in mitosis, what was happening? Again, they were forming a metaphasic plate. Yes. So the chromosomes will form a, what happened? The chromosome will arrange themselves in the equatorial plate of the, in the metaphase. Okay. Oops. Equatorial plate in the metaphase. Okay, sir. Yes, we got this. The chromosomes will get arranged and spindle fiber is gonna attached through this kinetochore. Yes, sir, we know that. Now you are able to understand. In the next phase, we will be having anaphase, right? We will have this anaphase, which is anaphase 2. And now these sister chromatids will also get separated. Now these sister chromatids, where they are going? Why I am not able to select them? Okay, yeah, now I am able to do that. Let me see here. Okay, now these sister chromatids are gonna separate here. Okay, in anaphase they are gonna separate. So how they are going to separate? See here, this one is going to go here and this one is going to go here. Similarly, this one like here, this one here, this one here, and this one here. Okay, done. See, both the sister chromatids is now separated. Now this was N, the whole was N, right? The whole was N, but now in this case this is N and this is N. Clear? Now they are going to move towards the pole and they're going to form this kind of structure. So one we had this and one we had this. From where it came? It came from during the separation of the anaphase 2. So this was N. And now since we can see that both the sister chromatids are separated and we can see 2 centromeres here. So we are going to say that now this is N and this is N. Clear? And this was a cell. Okay, this was a cell. Clear? The furrow formation is starting, right? Slowly they are going to divide in telophase and they are going to form N and N. Is it clear? So in anaphase 2, what is going to happen? Separation of sister chromatids. In anaphase 2, what is happening? Separation of sister chromatid is happening in anaphase 2. In anaphase 2, separation of sister chromatids is happening. This is what the cell cycle is, and this is how the meiosis 2 is happening. Clear to everyone? So we have discussed what? We have discussed the cell cycle in a very detail, and there are some points which we have not discussed, like quiescent stage and all those things. So what was quiescent stage? See here, if this is a cell cycle, okay, if this is a cell cycle, so in the cell cycle we had few stages like G1, S, G2, and then after that M phase. And after M phase, the cell again tries to enter what? The cell again tries to enter into the M phase. Oh, sorry. The cell again tries to enter into the, one second, I'll make a circle here first. Okay, yeah. So what did we had? We had in a cell cycle G1 phase, S phase, G2 phase, right? After G2 phase, we had what? We had M phase. And then after M phase, the cell again enters into the G1 phase. But what if the cell doesn't want to divide? So it exits the cell cycle, and this is known as G0 phase, which is known as quiescent phase. That means the cells which entered into the G0, that means they left their cell cycle. Now they're not going to divide. G0 is not a part of cell cycle. It is known as quiescent stage. It is known as what? Quiescent stage, right? And in this stage, there is no cell division. No cell division. Rather, the cell is doing all the activities, basic activities it is doing. Metabolically, cell is active. But the only thing which is it's not gonna do is that it is not going to divide. So, cell is not gonna divide, but it is metabolically active. So, this is what G naught. Okay. Now, sir, let us discuss about the significance of mitosis. Let's discuss about the significance. Sorry, meiosis. We discussed meiosis, right? Significance of meiosis. What is it? Now you tell. Where do we need meiosis? Gamete formation. Yes. Gametes formation. In the gametes formation we need. Now what, sir? If suppose you want to create a variation, So variation is created where? Variation is created in the meiosis only. Mitosis cannot create variation. So I can write here increases genetic variability. Increases what? Genetic variability. Yes or no? Yes, sir. Now chromosomes are number gets conserved. Sir, what is this? If I am writing that this line, chromosomes number gets conserved. How, sir? You are saying the very wrong thing. You just now said that 2n becomes n, 4n becomes n, then again you are saying the chromosome number is conserved. So let me understand, I'll make you understand about this concept that, see, this was Harry, okay, and this was Julie. Harry and Julie produced a baby. Harry was 2n and Julie was also 2n. They produced a baby which is again 2n, right? This is what we know. Okay. But let's suppose that for an instance, Harry and Julie decided that we are going to do mitosis when we are going to form the baby. Just they decided they are going to do mitosis while they are going to form the baby. Okay, sir. If during the mitosis, with the help of mitosis, if they are forming the gametes, so their gametes will be what? 2n and 2n, obviously. If they are forming the gametes through the mitosis, this is just imaginary condition, okay? This is just imagination. They decided to form the baby with the help of mitosis, okay? Mitosis. Now, the child is going to have what? The child is going to have 4n chromosomes. In a similar manner, their child also decided to form the gametes with the help of mitosis. Then, his child is going to have 8n chromosomes. That means what? That means we won't be the human beings. If a single chromosome, if a single gene gets little affected, mutated, lots and lots of changes happens. And see, it's just 2 and 4 and it is happening. The chromosome number will be constantly increased. So the species will what? It may destroy, right? So basically what is happening with the help of meiosis, we are conserving our chromosome number, right? We are conserving our chromosome number. How, sir? How we are conserving? See, with the help of meiosis, if they are forming the gamete, half of the number is gonna come, half of the chromosome is gonna come, and then they have 2n. Again, if their babies decide to do the meiosis only, so again they are going to produce n, and the baby's wife is gonna produce n, and again they are going to what? They are going to have a baby with 2n chromosomes only, right? So this is how meiosis is helping us to conserve the number of chromosomes. So this was all about the meiosis. And what was the difference between the mitosis and meiosis? Mitosis happens in the somatic cells, meiosis happens in the reproductive cells. Number of chromosomes is what? Preserved in mitosis. Number of chromosomes gets half in the meiosis. Okay. Meiosis happens for the reproduction. Mitosis happens for the growth and repairment. So this was all about this chapter, which is cell cycle. Now I am going to what? I am going to record a session for you, and in that session we will be discussing the whole cell cycle in just 10 minutes. and in those 10 minutes you will be able to revise that, but the whole topic. And on Sunday we are going to have what? We are going to have a quiz session at 11 o'clock. At 11 a.m. we are going to have a quiz session on this topic with top-notch questions. You're not going to see those types of questions anywhere. That's my promise. Okay, everyone. Till then, bye-bye. See you in